Hello and welcome to this video looking at Lac Operon and how it is involved in the regulation of gene expression. The Lac Operon is used in bacterium. E. coli normally metabolizes glucose as a respiratory substrate. However, if glucose is absent and lactose is present, lactose induces a production of two enzymes. And then we've got these so we've got some enzymes that are usually produced. I'm going to write them here. We have lactose, permease, which allows lactose to enter the bacterial cell. And the second enzyme is beta galactosidase. And this hydrolyzes lactose to glucose and galactose. So we can see how this is used, because if there was no glucose present, then we can use lactose to get the glucose. So the Lac Operon consists of a length of DNA, about 6,000 base pairs long, containing an operator region, which is here. This is our operator region. And we have the structural genes. This is Lac Z and this is Lac Y. And these are both structural genes. We also have, so these code for these two enzymes. So next to the operator region, we have the promoter region here. So these two here, these are the control sites. And then here we have got the regulatory gene, using the symbol I, which codes for a repressor protein. So we've got a regulatory gene, and it codes for the repressor protein. Okay. Right. So when this regulatory gene is expressed, the repressor protein produced binds to the operator, preventing RNA polymerase from binding to the promoter region, and the repressor protein therefore prevents the genes LAC-Z and LAC-Y from being transcripted or being expressed, so the enzymes are not made, so the gene is turned off. Okay. However, when lactose is added to the culture medium, once all the glucose has been used, molecules of lactose bind to the repressor protein molecules, and that alters the shape of the repressor protein, preventing it from binding to the operator. So the RNA polymerase enzyme can bind to the promoter region and begin transcribing the structural genes into mRNA, and then will be translated into the two enzymes. So lactose induces the enzymes needed to break it down. So that's just a brief overview, but we're going to look at it in more detail then. So we're going to look at, so the regulatory gene, we've already said, codes for that repressor protein. The promoter region is where RNA polymerase binds. It binds, RNA polymerase binds there, and it allows the transcription of the structural genes ZY. The operator region allows genes to be switched on or off. And the structural genes Z and Y, we've already said, they're expressed as enzyme lactose permease and galactosidase. So, when lactose is absent, I'm going to create a bit more room on the board so we can look at this. Okay. So when lactose is absent, the regulatory gene codes for the repressor protein. So we've got our repressor protein. And I'm going to just draw this in. So we've got our repressor protein. And the repressor protein, before this we've got some mRNA, and then that's going to go to our repressor protein. So the repressor protein is going to bind to the operator here. So it's going to bind operator region. So now RNA polymerase cannot bind to the promoter region. So preventing the transcription of the gene. So there's no transcription. So this means that LAC-Z and LAC-Y cannot be transcribed or expressed. And we have our enzymes not made. So the gene is off. Okay? So that is what happens when lactose is absent and glucose is present. Because you can imagine glucose would be the first option. So However, then have a look what happens when we have lactose present. When lactose is present and glucose is absent, lactose binds to repressor protein so let's just say we've got lactose and it's going to bind to the repressor protein and it's going to alter the shape of the repressor protein, kind of like how a non-competitive inhibitor does. It's going to change the tertiary structure of the protein and hence the active site that would have been used to bind to the operator region. The repressor protein and the operator region will no longer be complementary. So it's not complementary. If you're saying in a question that it's not complementary, say that it's not complementary to the operator region. So make sure you're being specific. So now the repressor protein cannot bind to the operator region. So RNA polymerase is free to bind to the promoter region. So then that will begin transcribing the structural genes to mRNA. So we've got some mRNA now. Structural genes. They're going to be transcribed. And we're going to be translated into two enzymes. So the lactose molecules then is broken down by the enzymes. Okay, that's everything for this video. Thank you for watching.